I think it's really amazing how little bacteria can create these fascinating cities. And also it's well organized, so you have like channels inside of these cities when actually they can transport the nutrients, they also can remove all the waste products, so this is really a complex structure. This is amazing. It's always amazing, especially when it's this like lorry that you start.
I'm an architect by training, but I now find myself in a microbiology lab, which is a bit unconventional maybe. But I think it's really inherently important that we as architects, designers are engaging with different disciplines and learn from different disciplines and also give something back.
The biofilm, it's really a wonderful structure. So it's a micro-architectural chef-d'oeuvre made by the bacteria. So I found really a strong link with designer and architects, because they were also interested by how bacteria will construct this little structure, complex structure. But also there is a part that is really interesting for example, material scientists, because we have a product.
In the field of architecture, we are wasting way too much energy in building materials, and becomes a serious problem. Usually we have to wait for 10 years, 20 years for a forest to grow, for a tree to grow, to develop. And with this method we generate cellulose, but in a much less time, energy and space-consuming way.
So it's like really a network of filaments that will help a net that will like hold the bacteria together, protecting them, but also like holding them together to a surface. When they grow as a community, as a group, they become stronger. We realize that the round one, they just basically lost the ability to produce.
If we understand these communities, microbial communities much better, we might be able to really extract information, extract methods, or extract any kind of strategies to our communities, and maybe we can learn from these things. The main idea was really to challenge with this artifact both disciplines. So we came up with a setup which involves a biofilm which grows, but it attaches during its growth to a set of threads. After 1 week of growth, we push the button and then the whole thing elevates. And that makes place, or like gives the opportunity for a new biofilm to emerge, basically underneath the old one. Three, two, one. It's basically layered mega cultures or mega communities. So we're talking about billions of bacteria really inhabiting these biofilms.
Say bye-bye. See you next week.
Bye. In this case, we really try to, in a way, if you will, collaborate with a microorganism to build something together. So it's not only about us humans, and also not only about collaborating between disciplines. It's here really collaborating, generating something, generating a structure, an artifact with a different life form.